On a molecular level, the basics of how genetic engineering works is every cell has DNA inside of it. The cell reads the DNA and it creates whatever the DNA says. So if the DNA says make this molecule, the cell will make that molecule. So what we as researchers do is we would type in what we want inserted into the DNA, then that would get placed within the DNA by enzymes and things like that, which you can buy. And afterwards, that DNA is placed inside of the cell, bacteria or other types of cells. And from that point on, it's a part of the DNA, so every time the cell replicates or is even around, it should read past that DNA and create exactly what we wrote. Now, to do it on an industrial scale, it's pretty much the same thing, except now they have tanks full of the enzymes instead of little bottles of the enzymes, and they have tanks of bacteria growing or whatever cell growing instead of little bits of cells growing. The biggest issue comes into purification, because anytime a genetically modified creature is, when it reads the DNA, it's not going to just create what you wrote, it's going to create everything else that's written on that piece of DNA too. So the real trick comes into, after getting a soup of stuff from your genetically engineered creature, how do you pick out the one thing that you want? Or how do you keep out the things that you don't want? And this really becomes a problem, say if you're using a cow as your genetic factory or genetic workhorse, and you want whatever item you made to come out through its milk. You definitely don't want contamination here or anything like that. So it's essentially, I would say, a three-step process where the people, the researchers, decide what to write or which molecule to create, getting that on a piece of DNA and getting that piece of DNA into some sort of a cell and having that cell replicate it. And that's, I would say, is the three-step process of how genetic engineering is done.